Okay, this is the 2023 multiple choice paper, and we're looking at the multicellular organism questions, starting at question 8. Okay, so role of stem cells in the human body is to produce what? Well, okay, let's have a look. Antibodies for defense. No, that would be lymphocytes. Lymphocytes do this. New skin tissue to repair a cut. Well, that'll work. Enzymes for digestion. No, that'll be pancreatic cells or perhaps salivary gland cells, something like that. And hormones for communications. No, that's going to be something like your pituitary or your pancreas for your hormones. So we're going to go for B. New skin tissue to repair a cut. These are undifferentiated cells that can differentiate into new tissue. Okay, which row in the table shows the functions controlled by each part of the brain? Okay, so we have the cerebrum here, the medulla or the brain stem. They're calling it the medulla here, so we better learn that because they haven't put brain stem in here at all. So cerebellum, cerebrum, medulla. I think this double L can catch you out as well, so just be careful with the names of these. Cerebrum and cerebellum are the two you're going to mix up mostly though, because they're very, very similar names. Well, the cerebrum, let's work down this list here, and cerebrum is for conscious thought or conscious thought. Okay, so it's going to be C or D, so these are both wrong, right? Medulla, breathing rate and heart rate, that's right. Coordination and balance is wrong, so that's right, so this has to be right, so the answer is C. Just work through a process of elimination. Start with the one that you're sure of. It's probably the easiest thing to do. If you're sure that cerebellum is for coordination and balance, start there and then work across. Whatever way you do it, just work in a process of elimination. Okay. Next question is, hormones are, what are hormones? Electrical messengers that travel along neurons? No, they're not. That's nerve impulses. Chemical messengers that travel along neurons, no, the hormones are nothing to do with neurons, so get that out of your head. But we do have chemical signals between neurons, so you don't want to mix them up. That's why they've put this in. They think you'll get mixed up in this. Electrical messengers that travel in the bloodstream, no, they're not electrical. And electrical messengers don't travel in the bloodstream. And D, chemical messengers that travel in the bloodstream, that's where you want a hormone in the bloodstream, and it's a chemical messenger. Okay. 11. Oh, I feel a calculation coming on. A man is fertile if his semen contains a minimum of 20 million sperm per centimetre cubed and at least 75% of the sperm cells are active. Okay. The table shows the result of semen analysis from 4 sperm samples. Identify the sample that was from an infertile man. All right, well, we're just checking off here. He must have 20 million sperm and at least 75% of the sperm are active. Okay. And we've got down here inactive sperm. This should be easy enough. So the only one that has less than 75% inactive sperm is this one here, because take 30 from 100, you get 70% inactive sperm. And it does have a minimum of 20 million sperm cells. All of these do, so this is your answer B. Okay, so all of these check out, and these are inactive, so you've just got to watch out for the active, inactive. Okay, so what did we say? This is it here, B. All right, in mice, the dominant form of one gene, big B, determines black coat color, and the recessive form, little b, determines brown coat color. If 2 heterozygous mice were crossed, the expected phenotype of the offspring would be the phenotypes. Now, watch out for this, not the genotypes. Somebody's handily done this. Somebody's seen this, used this paper before for a bit of revision. Anyway, they've done the Punnett square. Big B, little b, big B, little b looks good. And what have we got? We have 3 to 1. So we're going to have 3 of big B, which is black coat. And we're going to have 1 brown. Okay, the recessive is brown. Only one of these is going to show the phenotype of brown, because the others all have a dominant allele. It's definitely dominant. Yep. Okay, so 3 black, 1 brown. Answer is A. There you go. Nice and easy. Hey, we've got the whole leaf. We've got the whole leaf. This is a good. This is just a good revision diagram. You should just learn this. So this is how they show the leaf in National 5 exams. The diagram shows a cross-section of a leaf. Yeah. Okay. So we've got cells up here, we've got cells here, we've got cells here, and we've got one cell down here. Okay, so we've got the upper epidermis, the lower epidermis. We have some kind of cuticle along there. We have palisade cells, and we have spongy mesophyll cells, right? You should be able to identify all of those. Just learn it. It's the only way. Which row in the table identifies the parts of the leaf? Well, we've got the upper epidermis is W. Well, palisade, this is at X. So that's correct. Palisade is correct. And that's correct. Okay. So they're wrong and wrong. Spongy mesophyll is Y. So that's correct and that's correct. That's wrong. That's wrong. Upper epidermis is W. So that's wrong. And that's correct. And we have hit on the answer before we get to guard cell, which is the last one there. And that is D. Answer is D. Okay. So can't fit all that in my screen. But we've got all of the cells. That's a good diagram. I would sit down and learn that diagram. Upper epidermis, palisade cells, spongy mesophyll, guard cells, and lower epidermis, if you want to put that in. Plus a bit of cuticle and top waxy cuticle. Okay. Question 14. Oh, this is a big one. All right. An investigation was carried out to compare transpiration in two different species of plant, G and H. The diagram shows the setup to measure transpiration in species G. Okay, species G is this sealed polythene bag around the plant pot, balance, and any water in the pot's going to go out through the leaves, right? Can't get out through the plastic because it's a sealed polythene bag. Okay, which diagram shows the setup for species H, which would allow a valid comparison in the rate of transpiration of the two species? Well, for a valid comparison, you must keep everything in the experiment the same except for the thing that you're testing. In this case, we're testing the species. So we want to find a picture that looks exactly the same as this, except that's going to be an H, not a G, right? So that's wrong because the plastic bag going right over the top of the plant. This looks good. That's wrong, no plastic bag. And okay, we have two to choose from here that look right. What's the difference between these? Well, the difference between these two is the number of leaves, right? 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5. All right, and this one up here has 1, 2, 3, 4, 5, 6, 7 leaves. I'm going to go for D. I'll have to check that and make sure it's right. But I think it will be. It's the same number of leaves pretty much, or possibly the same leafy surface area. Okay, so you're trying to get the conditions to be exactly the same, and the only thing we want to change is the species. Species G up here, species H down here. I don't see any other difference that's valid. No. Okay, so validity is keeping everything the same except for the thing you're testing. Right, we've now got two questions. Right, we now have two questions that were a bit challenging for the students on that particular exam. 2023, which row in the table describes some features of veins, right? Remember, veins have valves. You just go to bed saying that little mantra to yourself, veins have valves. Okay. Direction of blood flow, blood pressure in the vein, width of the central channel. Okay, nothing to do with valves. So even though we learned veins have valves, it's not mentioned. Okay, so let's see. Direction of blood flow. Veins carry blood towards the heart, right? So that's correct, and that's correct, and that's wrong, and that's wrong. Okay, blood pressure in the vein, low blood pressure. That's correct and that's correct. That's wrong, but that's already wrong. Okay, so we're going B and D. And wide channel is correct and narrow channel is wrong. So we're going to go for B. Quite a few students got that one wrong, according to the report, the course report. Let's have a look at this one. And this one, also I put the little exclamation mark here. The diagram shows a villus. Or a villus. Okay, identify the nutrients from food that are absorbed into the lacteal, or the lacteal, which is this grey bit here. This is the part of the lymphatic system, and it's all oily fluid, and it absorbs fats and glycerol, right? Now, why fats and glycerol? Why do they talk about fats and glycerol? And you may not have learned this in National 5 because you just learned that glycerol and fatty acids are absorbed. But in fact, a The fats that exist in I'm doing this very rough. The fats that exist in living things, so the fatty tissue that you eat, the beef that you eat, the lovely fatty rind on it or whatever, these things are called triglycerides. You've got 3 fatty acids, 3 times fatty acid, and 1 times glycerol. The whole thing is called a triglyceride. Triglyceride. Okay? So glycerol with 3 fatty acids attached, right? Basically. So that's why they talk about glycerol and fatty acids. If you didn't go into that detail in National 5, it's probably just worth going into that detail a little bit, because it reminds you that glycerol and fatty acids are both of these things that are the fatty stuff that gets absorbed by the lacteal, and we want glycerol and fatty acids. Now watch out, glucose and glycerol look similar sort of words, but make sure you got them clear in your mind. Glycerol and fatty acids, what we want, not amino acids. Fatty acids, amino acids come from proteins and they're water soluble. Fatty acids come from triglycerides and they are fat soluble, oil soluble. Okay? Any more? Okay, 3 students carried out an investigation into the effect of exercise on heart rate. Each student measured their heart rate after completing the same exercises. After each student's heart rate returned to its resting rate, they repeated the process. Which of the following would increase the reliability of the results? Remember, reliability is a question of doing more repeats. Do the experiment over and over again. There's lots of human error in biological experiments, plus there's lots of subject error because individual organisms are all genetically unique. So what we want is lots of repeats. So let's see, increase the time spent exercising. Not really. Change the exercise location. Not really. Change the type of exercise each time. No, that would be invalid. And increase the number of students exercising. Well, increasing the number of students exercising will give us more repeats of the experiment. So more data. More data is good for reliability. I'm going to go for D. Okay. Right, in an ecosystem, that's us. We're done. Multicellular organisms up there. 2 question 17. We'll go on to the ecology questions another time. Hope you enjoyed the video. See you next time.